Hello, my name is Rebecca Erwin, and I'm a junior in chemical engineering. I'm going to be discussing my research on designing advanced membrane materials for produced water treatment. In industry, separating mixtures is a common problem that is encountered, whether that be separating products from a chemical process, raw materials, or wastewater that is to be either omitted into the environment or purified for drinking. These separation processes are typically thermally driven and are both highly consuming of energy and costly. Our research specifically focuses on separating hypersaline water that is generated from oil drilling and fracking. We propose that membranes are a promising alternative to thermally driven separation as they are both energy and cost efficient. To understand how membrane-based separation works, we just have to imagine things that we encounter in our everyday life, such as coffee filters or masks. Membrane-based separation works by a semi-permeable barrier that only allows for certain small molecules to pass through. It is selective based on molecular weight. Commercially, this process is driven by reverse osmosis, where hydraulic pressure is the driving force that is pushing smaller molecules across this membrane. This is a very successful process in separating salt water to purified water like seawater. However, this process is limited by salinity and is not very successful in separating hypersaline solutions. That is why our group is working on designing forward osmosis membranes to better separate hypersaline solutions. Forward osmosis works by creating an osmotic pressure gradient, where across a membrane on opposite sides there is a feed solution and then there is a draw solution. Now the draw solution is at a higher concentration than the feed solution, and as a result water diffuses across this membrane to exist in the draw solution. This is a very successful method as no hydraulic pressure is required for this process. Now, comparing forward osmosis and reverse osmosis membrane morphologies, we first need to look at general membrane morphologies for separation. Generally, there is an active layer, and that is responsible for primarily determining what diffuses across this membrane. That is fused to a support layer, and this is responsible for supporting the active layer. In reverse osmosis, it is desired that the support layer is thick and sturdy to withstand forces of hydraulic pressure, but for forward osmosis, we want this layer to be as thin as possible to facilitate speedy diffusion and maintain this osmotic pressure gradient that achieves a high water flux. So our group is working on designing better support layers for forward osmosis by the method of layer-by-layer deposition. Now this is achieved by layering oppositely charged polymers to one another through a process of dipping and rinsing in DI water and repeating with the oppositely charged polymer, rinsing and repeating. This is an iterative process that can be repeated until the desired thickness is achieved. Now once these layers are formed, there's a post-treatment that occurs where acidic immersion, rinsing, and drying creates and breaks bonds and reforms to create this porous structure. And as a result, porous structures are achieved. Now, these porous structures can vary significantly depending on many variables. We specifically are varying deposition time, polymer molecular weight, and the specific pH of the acidic treatment and post-treatment. And with manipulating these variables, a wide spectrum of varying porosities, thicknesses, and morphologies can be achieved. Once we create enough membranes, we're going to be evaluating how these membranes behave through both batch testing and continuous setup testing to evaluate how these membranes work in both steady and unsteady state. In the future, we hope to test these membranes and see how they perform on industrial hypersaline water by matching the same concentrations and temperatures that occur in industry. To conclude this presentation, I want to acknowledge the funding for this project, which was by the National Science Foundation EPSCoR project. And I want to thank everyone for watching and welcome any questions.